Welcome to Conversations That Matter. Joining me now is Mike Minier. Mike, who's our guest today?
Stu, today we have Corey Nislow. He's a professor at UBC with the School of Pharmaceutical Sciences. He is doing some very interesting work that I think we can all relate to. We've probably all either gone to the doctor or had somebody that's gone to the doctor, and then there's this long groping along to find the right medicine that will treat whatever it is you have. Well, through genomic research with Genome BC, UBC, and the BC Pharmacists Association, they're actually coming up with a pilot study that could bring that highly inefficient, painful process we all go through down to something that's much more manageable. And really, it's this concept of personalized medicine, and it's all using genomic tools.
Well, he says we're right on the cusp of a dramatic change, a very exciting time in the delivery of medicine. And it's happening here in BC ahead of anywhere else.
Well, you know, we've all watched over the last 10 or so years the outcomes of the mapping of the human genome, and we all have these high hopes for what could happen. And this is actually where everyday folks, all of us, could see some very dramatic changes in our medical care and our regular lives, because they'll be able to take a saliva sample and find the medicines that are going to work just for you, and find out how long it's going to take them to work, and cut out a lot of the, you know, as I said earlier, the discomfort and the inefficiencies of medicine and pharmaceuticals today.
It's quite extraordinary, the concept of being able to take a sample of your saliva, sequence your DNA, and then be able to figure out what dosage or drugs you should have.
It's remarkable stuff, and I'm still trying to figure out how to use a cell phone.
Well, Corey is an incredibly enthusiastic proponent of this, and he's leading the research on that. Let's go to that conversation now. Thank you for joining me today.
Thank you very much, Stuart.
I think that we're at a really exciting time in the mapping of the human genome and what it's going to mean to us as the delivery of individual medicine. Tell me a little bit about the work that you're working on right now.
Absolutely. So before I start, I can't echo what you just said more emphatically. 2015 is going to be the year that genomics gets real. In my humble opinion.
Gets real.
It gets real.
And how's that going to play itself out?
Well, so we've known the sequence of the human genome, one human genome, since approximately the year 2000. So we're coming on 15 years. Now, in 2015, we have the sequence of probably 1,000 individuals from all over the world. And
that's it? There's only been about 1,000 people who have been sequenced so far?
Yeah, fully sequenced. Yeah.
Huh.
That is really, we are on one side of a tipping point. And the reason I'm so enthusiastic about 2015 is that we're going to see several thousand individuals sequenced.
So what has happened that allows us to go from 1,000 in 15 years to thousands in the space of 1 year? What are some of the components that had to come together to make that possible?
A lot of things had to independently come together to make this somewhat audacious prediction possible. During the interval from the first human genome being sequenced around the year 2000, during that 15-year interval from 2000 to now, the costs and the resources required to sequence a human genome decreased 1,000,000-fold.
What's the benefit, first of all, to the system in knowing the genomic sequence of you versus me versus the guy over there operating the camera?
Well, let's break it down what that means. If you come into our shop at UBC and I sequence your genome, or you don't have to come in, you can just spit in a tube and put it in the post. After I sequence your genome and compare it to all the other genomes that have been sequenced, and the comparison is key, then I'll be able to come up with approximately 3 million places in your 3 billion in your genome, which comprises 3 billion letters, I'll be able to come up with 3 million, or 0.1% of your genome, that makes you Stu, compared to the 3 million that make me. And
okay, I can understand why I want to know that, but
Right.
why does the system, like the healthcare system, want to know that? What's the benefit to them?
well, the benefit to them, and by them I mean to all of us, is within those 3 million differences, we're going to find differences in genes that encode proteins that are critically important for how you respond or don't respond to a particular medication. And to say it really simply, some of your genes will have variants, and they're not mutations, they're just normal variants that contribute to our individuality. Your genes will encode variants that will make you metabolize some drugs quicker than others, make you metabolize some drugs slower than others. Some of the genes will encode receptors that will bind to drugs, and in some cases won't bind to other drugs. So just at that very simple level, which is a burgeoning field called pharmacogenomics, which is the combination of pharmacology and pharmacy with genomics.
You're working on a pilot project on that right now.
We are again, this is part of the reason why 2015 is the year when genomics gets real, because we have we've been working on a partnership with BC Pharmacy Association, and in 2015 the project is going to officially launch. We're going to start enrolling patients, and pharmacists are going to be the individuals who are taking samples from clients and patients who come to pharmacy, sending the samples to us, and we're going to be decoding the genome and seeing and asking the question, what in this individual's genome can inform what drugs they should or shouldn't be taking, and can we learn anything about starting doses?
Is that where the greatest benefit in understanding our individual genome lies, in recognizing what our own interaction with medicine is going to be?
You know, scientists use this phrase called low-hanging fruit, and it has, for some people, has a negative connotation, like we're lazy and we're just going after what's easy. But it's actually what's practical. Right now, the genome can inform us about what drugs you should and shouldn't be taking. That is a well-established fact. It's not controversial. Doing that is not very expensive, and so it is low-hanging fruit in the sense of it's something we can do now. And more importantly, it's going to set the stage for things that are more difficult to do, things that are harder to predict based on your genetic endowment.
But immediately, I can't help but think, good, I want to know which drugs will or won't interact with my genome positively or adversely, so that I have better health outcomes and fewer risks.
I think that's again, it's absolutely non-controversial. Why wouldn't, if one could, why wouldn't one want to know that I shouldn't be taking this drug because it can't help, and all I can experience are side effects? And if there's another drug with a similar mechanism, but that acts slightly differently, that could help, why wouldn't I want to know that? And furthermore, if
Well, you're not going to get an objection from me.
Right. Yet, so this is there's no controversy, but there's a conundrum, and that is, why aren't we doing it? We've known this for a decade. We didn't need the full genome sequence to tell us this, but why aren't we doing this? And you'll probably get as many answers as people you ask the question, but I would say a big part of the reason we're not doing this is there hasn't been a healthcare profession that stepped up and said, we're going to take this on. Cardiologists are interested in prescribing you the right heart drug for your particular condition. But typically, and this is not something I'm making up, typically they do not look into your genetics of your drug metabolizing enzymes to inform the dose and particular drug they're going to prescribe for you. And part of the reason is there's no one pushing them to do that. It's not their fault. It's not an oncologist's fault that he's not taking in your genetic repertoire of metabolizing enzymes in thinking about what drug to prescribe. But for the oncologist, when the drugs start costing $100,000 for a 6-month course, and now we're talking about very serious amounts of money, and whether or not you metabolize that drug is going to have significant monetary consequences, we're going to have to start looking at that.
You have my mind reeling at the moment because I hadn't thought about it as the gatekeeper to moving forward would be finding the right group that would make sense for them to say, we want to look at everything.
So, because you and I think are in agreement here that it's a rational, logical thing to do. But often that's where conversations end. Okay, now who's going to do it? And so 2015, we're going to start walking the walk. And we've been talking about personalized medicine, not we, but physicians have been talking about personalized medicine, taking the patient as a whole, as an individual, and tailoring the medication for that person. I mean, they've been doing that since Greek physicians, right? But now we can actually do it. So the question is who?
Well,
and I would say, based on, I've learned an enormous amount over the last year about just how many different pieces of patient information the pharmacist can survey at once. And these are the individuals who I would offer are in the best place to do it.
Yeah, I was talking to a pharmacist last night as I prepared for our interview, and he was sorry that he wasn't part of this program. He said, we didn't get picked. He said, but I'm really excited about what the opportunities are, because as he pointed out, every day I'm dealing with people and I'm giving them the drugs that have been prescribed to them. How beneficial would it be to them if I can look at their file and say, okay, I will give you this formulation or this maker's version of that drug because I know that it's the best for your genome. And I can even go back to the physician and say they don't need that much, they can take less.
Yes, and a big part of this is, so we've identified, I think, a very qualified professional to serve this role. An important part of their role is going to be the educational part. And that is, we now, as we're having a conversation, every interaction that a pharmacist has with the client can be a conversation that has a small educational component about what we know today about how your genes interact with your environment. In this case, the environment being what drugs you're going to be putting into your body. And every one of those interactions can be a positive, non-hyperbolic, down-to-earth discussion of what we know, what we can do, and what we can't do yet. And so this idea of having a conduit of all of your health information through the pharmacist to the patient, and the conduit includes that educational component and dampen down any potential concerns or fears, is I think an incredible opportunity.
walk me through the process. How are you going to be able to take this from what is still a concept through a pilot project and actually make it work? Like, what are the components that will get my DNA or my genome to you so that you can get this started?
Okay, fair enough. This is, you walk into, I'll be a pharmacist today. So you walk into the pharmacy. For the purposes of this study, I ask you if you're interested in reading some information about a pilot project that we have ongoing with UBC, Genome BC, and the BC Pharmacy Association. If you agree to read the information and you like what you read, basically the information just describes the scope of the project. And I'll go into that in a little bit more detail in a moment, but let's go walk through the process. So you come in, you agree that you want to make a contribution to your community and society by providing a sample. I give you a small tube. You work up some saliva for about a minute or two, spit into that tube, hand it to me, I cap it, rock it up and down. That inactivates anything in your spit, like anything that's potentially infectious, and your DNA is stabilized. One nice thing about DNA is it's probably one of the most stable molecules that we know. So I now have your sample.
Yeah.
The tube is barcoded. Your name and the barcode are only associated at the pharmacy. They're delinked when the sample is sent to my lab at UBC.
Why is that? So that you won't know who I am?
That's exactly right. I want to know that this sample belongs to this barcode. I don't want to be able to go back and link it to you as an individual. And
so that if we're at a meeting at some social event or a meeting, you don't want to go, oh yeah, right. I just
that's the guy with the wacky gene.
Right.
But there's privacy and ethical and legal issues that are very important to work out. But in order to show that the logistics are possible and that the science is possible, we are going to take those ethical, legal, and social issues and just set them right here, just off to the side for now.
There's a firewall there.
There's a fire there, absolutely, multiple levels. So your sample comes into the lab, and our technicians will process it. The entire, this is fairly standard molecular biology that follows standard operating procedures. So if we take your sample at UBC, or if we took it at a lab at Stanford, or in a lab in Adelaide, Australia, the result would be the same. But it comes to UBC, we process your sample, we chop up your DNA, which, by the way, if you stretched it end to end, would be 1.8 meters long. So
that's 3 billion letters.
It's 3 billion letters, and it's a big molecule. We chop it up into very small, manageable pieces. We add some small DNAs that allow our sequencer to decode it. Now you have essentially the world's largest jigsaw puzzle of your DNA in little bite-sized fragments. We put that information into a computer. The computer does, imagine a humongous game of Tetris where the pieces are just lined up and built on top of each other until we can reconstruct that string of 3 billion letters. Then we take that, we compare it to all the other genomes that are in the database, and lo and behold, most of the time, if you are who you are, your genome will be 99.9% similar to all the other genomes.
Around the world.
Around the world. Except, as I mentioned, you will differ in about 3 million places. So in about 1% of your DNA will have variance compared to the reference area.
So coming back to pharmacogenomics, did I say that correctly?
Yes.
Yeah. You're now armed with this information. What do you do with it?
Okay, so in the course of this project, we're going to collect 200 human samples. We're going to sequence the exomes. That's the protein, the fraction of your genome that contains genes, which is actually only a small portion of your genome. We're going to collect that information. We're going to independently take that same DNA and perform a traditional simple PCR test that's approved to identify drug metabolizing variants in your genome. That will allow us to compare how well our DNA sequencing test does relative to a benchmark that's approved by Health Canada and the FDA. And then we're going to run a number of in silico or in computer tests to ask, with this individual's genome, what should have been their dose of drug X, Y, and Z. In particular, we're going to focus on a drug called warfarin that's given for anticoagulation purposes.
So you're starting with one drug.
We're starting with one drug, but more importantly, we're starting in silico, in that we're going to run the experiment and ask, what would we have done if we were to go inform the patient as to what their dose should be? Now, there's a lot of woulds and ifs in that sentence, and the reason is that the focus of our study is the logistics. And that may not seem super sexy, but it's absolutely critical because we have to make sure that we get the process right for when we do go and make a recommendation, that we've looked at all the possibilities for error and noise and minimized all of them to the best of our abilities.
Be realistic here. How long before this starts to work its way into our daily lives here in British Columbia?
There's this quote that's been attributed to everyone who's ever done anything good in Silicon Valley. The quote goes something like, people tend to overestimate what you can do in 1 year and underestimate what you can do in 3 years. And I think it's absolutely appropriate for our project.
It's, going back to the beginning of our conversation, you said this is the year. And having gone through this last 40 minutes of conversation with you, I believe you that this is the year. Thank you very much.
Thank you so much.
And all the best in the work that you're doing. I think it's extraordinary.
Thank you. Thank you.